But it's an absolute pleasure to be here. I'm going to talk to you guys today about history, about interconnections, about old-growth forests, about fish and salmon and trees and fungi. I'm going to talk a lot about myself a little bit, actually. Tell you my story, the way I came to do this work, the way I came to think about these things, and what I've been thinking about and starting to think about now. Because I just finished my first week, as Anne said, in a new job with the Canadian Forest Service in Victoria. And I'm still not sure what I'm doing precisely, but it's something forest degradation. No one really knows what this means, and we're going to talk about this in a second, but sort of the dark side of forest health, right? That's okay. Let me just, hold on a second. Yeah, I know it's being recorded. There we go. First of all, you folks, thank you very kindly again for inviting me. I didn't somehow know who Nature Vancouver was. But I read your wonderful website. And one of the hats I wear is I'm a director, a little bit of self-promotion here for the Forest History Association of British Columbia. And please join us, check us out. We also have talks and things. But we're very interested, especially in local histories. So reading this brief history of the BNHS by John Roththorne on the bottom was really fun last night while on my ferry ride over.
Do you want to make friends with the mic?
Yeah, I can. Is this helpful? Okay. So, yeah, I was shocked and amazed to hear about all the amazing things you folks have done over the last century and how deep the history went. And I guess it was just about 100 years now. And one of those things, of course, is protecting a lot of the forests around Vancouver. Protecting old growth right now, what we call old growth anyways. Oops, we're back. This has been in the news, right? The things Vancouver, what were you, Natural History Society was doing prior, is suddenly happening all over the province. And it's a good thing we were doing it in 1920. And it's really fascinating to me historically that it's 100 years ago these kind of movements began. And now we're having kind of another wave of, hey, there's not much of these places left. So of course we have, one of the other hats I wear is becoming a forester. And foresters love all their acronyms, right? So all of a sudden we have these OGDAs and OGMAs, old growth deferral areas, old growth management areas, on and on. And so it's been really interesting to me that these discussions are happening, and really important to me that we start talking about them collectively. And citizens and citizen groups like this get involved, and of course, we're the ones pushing this. So the Vancouver Natural History Society, I was shocked to see this as your name, because one of my kind of side projects is I call it Make Natural History Cool Again. Because as a scientist, it's been kind of poo-pooed. It's out of fashion to be a natural historian. They look down their nose. Scientists want data now instead of stories. And we want hypothesis-driven experiments rather than observation. So one of the kind of goals of this talk for me is to articulate that I think stories are really important, and that citizen science is a really effective way to get those stories about place. So my new work, I'm thinking about this question: what is a healthy forest? It's really easy for us now to think, as an analogy to people, what a healthy person is. But it's pretty hard to define scientifically. We haven't found yet, at least from a Western context, the health hormone or something that healthy people have that, like, yeah, you did it, now you're healthy. So And it's even harder then with a forest. What does a healthy forest look like? What are its characteristics? Can we say a forest is healthy if it's our goal to promote forest health? Similarly, and I'm not sure if this is true, this is a genuine question for me, and I think I put my email on the bottom here. If any of you have any ideas and opinions, what is forest health to you, and what is an unhealthy forest, please let me know, because this is something I'm going to be thinking about hopefully for the next chunk of my career. In Western medical health, we do usually can tell when someone is sick. A forest, it's a little harder. Right now, we have a lot of challenges in our forests, from fires and climate change, droughts, to insect predation of all kinds, and also fungal predation. We're going to talk about that in a little bit. So these are really genuine questions for me. And I'm going to present to you kind of two hypotheses I have right now, or I think three actually. So I'm going to say, just off the top of my head, healthy forests are ones that have complexity and attachments somehow. There's some depth to their being. And the other side of that then is healthy forests are forests with relationships, right? And of course, if you have relationships, usually healthy forests are ones with history. And I think this is something really important because in science and forestry, we think often about a lot of the models up until now have been equilibrial-based models, we call them, or steady state, or all of the arguments will be like, okay, in 200 years, our forests are going to be like this for the rest of time. And that, of course, isn't what we see. When we have our actual forests that we know and love around us, what matters is their recent past, right? Their natural history. If you go to the North Shore and walk around, what do we often talk about to understand the forest? Or even closer to home here, the Pacific Spirit, the immediate history of logging and so on 100 years ago and fire, that shapes the forest a lot more than anything else, right? So I'm going to talk just for a second then about my history. I was born in this wonderful city, Toronto. It seems like a strange place for someone who would grow up to love forests. But of course, Toronto's full, it's a city of ravines and greenery. And if any of you have been there, I grew up kind of riding my mountain bike along these systems of connected pathways. And of course, along these connected pathways come birds. Despite living downtown, I frequently would see coyotes and foxes. And this is what it looks like. So this is not far from where I grew up in Riverdale. And that's downtown Toronto on the, I don't know, right? The right-hand side. So incredibly green city. One of my dreams, incidentally, is to see the highway there on the left, the Don Valley, be eliminated and return the river valley to the trees and the animals and the children. But I grew up paddling a lot of time in Muskoka in the forests of southern Ontario. And eventually I came of age and left my sad mother and father and came here. So I actually did my undergraduate at University of British Columbia. And does anyone know Science 1 at UBC? Yeah, so I came here and was in this really special program at University of British Columbia called Science 1, which was the best year of education in my life. And it really pushed kind of integrative thinking, collaboration, and exploration and curiosity-driven science. I was fortunate enough then to work in the electron microscopy lab. I didn't get to play with this machine, this electron microscope. Instead, I washed dishes. But eventually, I did get to play with it. And I did some work with some scientists named Patrick Keeling and Brian Leander on protozoans and algae. So they're very, very small. And so these pictures here, this is volvox, they're called. They're one of my favorite algae. And they're one of the early, they're a model organism anyways for multicellularity. So each of these little dots is a different cell, and they all get together to make kind of one big cell. They're really, really interesting. They grow in puddles and stuff. I also was fortunate enough to work with a woman named Leticia Aviles on social spiders. So these are very, very tiny spiders, but together become the thing of nightmares. And their webs take entire trees and groves of trees. And they demonstrate this incredible behavior where, like most spiders, their eyesight is poor. And so they build these collective webs and need to feel their prey. But of course, you can't feel your prey if there are other spiders moving around and disturbing the web. So what they do is they all will be moving randomly, and then if they detect prey, they'll all stop. And then they'll all move. And then they'll all stop again. And they'll jerk, and you'll see hundreds of them moving like this, slowly converging on some poor writhing dragonfly or whatever. So really incredible, and that kind of behavior, seeing that, observing that, really got me thinking about collective behavior, complexity, how things work together to do things. I went on, I came back to Canada, to McGill University in Montreal, and I was fortunate enough now, slowly moving towards the land of plants, to work on this really chonky science. So this is in Cedar Creek. And each of these little squares, this is a multi-million, hundreds of millions of dollar experiment, ran for many, many years. Each of those squares, and those are, I think, 10 by 10 or 20 meters by 20 meters, are plots where they'll have different numbers of species. So 2, 4, 8, oh, they have monocultures as well, and up to 16, I think, species of plants in different mixtures. And the question they were trying to ask, this is in the late 90s and early 2000s, is why was diversity good? And that's something we think about all the time. We talk about diversity, biodiversity, we assume is good, we want it for all sorts of reasons, but they were trying to really answer, like, can we, as scientists, find the diversity hormone or whatever? And these are two of their key pictures they were thinking about. So after many, many years and hundreds and thousands of hours of undergraduates counting plants and weighing them, this axis here is ecosystem stability, and those x-axis there is the number of species. So as you get more and more species, the line goes up. The system becomes more stable. And this has really become kind of a real driving idea of why biodiversity is good. But there's another graph there that I find is really, really interesting. And so this is the same y-axis, stability. And then the x-axis is also the same, the species number and the treatment, but the line is, instead of the stability of all of them weighed together, it's the stability of each individual species. So this is really, really interesting. So it means that diversity is good for everybody, but sometimes for particular species, there's a lot more competition. Maybe you have good years and bad years. It actually gets harder, or at least more variable, when there's more species there. So anyways, I did some stuff with that. I'm just going to skip over that. I decided to go traveling again. I went to this incredible country, the Republic of Georgia. It's in the news again because Russia's involved. I was there in 2010, and Russia invaded the previous time in 2008. So I was there to teach their kids English because they wanted them to learn English instead of Russian. It's a land of monasteries and history, and song and poetry. So here, there's a younger me of the class, and it was the first place that I'd been where there were songs and stories embedded in the land from the people there. And archaeologically, the Georgian people have been there, as far as we can tell, in many respects, as back to the last ice age. So a very, very long time. Does anyone know? Has anyone seen this map before? Any guesses?
It's the stands, isn't
It stands.
it? Stands.
Like the Afghanistans and those things.
Afghanistan.
Not
It's
quite.
also got the west coast of Vancouver.
Uh-huh. What are the green things?
Forests.
Yeah. So one day I was looking at this map, and I didn't know what I wanted to do with the rest of my life. I was young and finished traveling, and I saw this map, and I looked at it, I'm like, wait a second, I want to go to all these places. I want to go to Chile in the south of South America. I want to go back to Vancouver. I just finished being in Georgia, and there's a little bit of green around, what is that, the eastern side of the Black Sea there where I was. So this is a map of all the temperate rainforests in the world. There's a lot of places you wouldn't expect if you didn't study this thing. Northern Norway, Wales, Ireland, of course, we think of as green, had many trees a long time ago.
Ireland's all
Not in Africa. Interesting.
done fir plantations now.
It is all tech fir plantations now. They grow like stink there, though. And for good reason, the climate, well, a lot of reasons, but the climate's different. There's also fewer fungal pathogens. This is the theory, anyways, that they have here. It's a whole nother talk. So that brought me back here, finally, to the Great Bear Rainforest. And I'm sure many of you have been there. Beautiful land of mist and rain. This is me and my colleagues. We're doing work along salmon streams. And we know this story. The salmon come back, the bears eat them, nothing happens. And the salmon die, right? And then they're dying, they get, the bears cast them through the forest, and they rot on the land, and their nutrients in their flesh goes into the soil. So this is a site that I was working on. The salmon are swimming in. Bears eat them, and of course, more importantly, lots of things that we don't normally talk about eat them. The biggest consumer of the salmon are these flies, of course. And so you can imagine in the fall, the forest, and you stumble upon a small area with tens or hundreds of salmon, and they're just writhing with maggots. And out of the maggots, of course, come adult flies. And so what I was initially studying here, I was volunteering, this little bird. And of course, we're all birders here. This is one of my favorite birds, the Pacific wren. And so the question that I was working with, his name was John Reynolds, a professor at SFU, and his graduate student, Marlene Wagner, their question was kind of how deep does the salmon nutrients flow? We know it feeds the bears, we can watch that. We know it feeds the flies, but does it also feed the insectivorous wrens? And so we were asking a lot of questions about that, and furthermore, does it help the wrens? Does it increase their nesting success? So the wren became one of my favorite birds. Part of that job, I don't know, as we're all birders, so enough birders here, was trying to find wren nests, because part of learning their nesting success would be counting how many eggs they have and following the success or lack of it of their young. That was objectively the hardest job I've ever had. I was terrible at it. You're finding a wren nest, which is a little ball of moss in a world of balls of moss. So this is the picture then. We had the forest, we had the wrens, we had the fish. And of course from the fish then we have the ocean. And we're going to try and draw some of these connections. Indirectly then, I have one little dotted line, but the ocean, of course, affecting the forest, it's affecting all these other things. My idea then was, well, if this is going from the fish to the trees, it's got to go through the soil first, right? If it's going through the soil, it's got to be going through the mushrooms, and through the mushrooms to the trees, because we all know about mycorrhizal networks. Now it's suddenly famous. We didn't 10 or 20 years ago, or at least it wasn't popular knowledge. And then, of course, what I call the 10,000 things, right? Literally everything in these systems that we've measured, as far as I have read, has shown evidence of salmon nutrients in their flesh and bones and feathers and hair. Here's a drawing. I'm going to skip over this, but these are all the things that in the literature we've looked at least when I put this together a few years ago. And of course, there's something really important in the bottom left there. is the health sick people who've been there for very, very long and are often brushed over in this kind of literature. And it's not just the health sick, this has also been repeated in Alaska, anywhere we look at that, but all of these places, of course, have had people for a very long time also influencing all of these things, from the birds and the eagles, to the ocean, to the fish, to the soil, to the trees, to the ravens. So then there's me, Johnny come lately, showing up, bright-eyed and bushy-tailed, with all these ideas. And of course, something that scientists do is we pick usually one little thing to look at. So in this case, I'm looking at 3, which is already profoundly interdisciplinary and turned out to be really, really hard. But of course, we only look at it for a short period of time. We usually only look at it in the summer. We only look at it in the daytime. And the longest most university studies go would be a PhD, which would be 2, 3, maybe years of field work, right? So this is another appeal that I have to the importance of history, is that it can look at these things on another scale of time. And of course, part of history then is acknowledging there are no new ideas in science. So there's this wonderful book by Paul Kroger, local from Vancouver here. And of course, he wrote about this in 2012, and people have been talking about this for a very, very long time. To give some context, this is what it looks like. You have the dead fish turning into a skeleton. That process can take a few days to 2 years. And part of that is the nitrogen, phosphorus, calcium, and other nutrients go into the soil from the fish. So my approach here is we're going to get into the chunky part of the talk a little bit. But I'm going to try and break it down so that the science side of things is I really looked at 2 types of data. I looked at the whole region, and I compared streams that have no salmon, some salmon, and a lot of salmon. So I call that salmon density. There's the salmon density gradient on the x-axis. And then the y-axis would be whatever I'm measuring. My other data set was above and below waterfalls. So I think I had 5 sites with waterfalls, and I would look at a single site then, below the waterfall, where there was salmon, and then above the waterfall, there wouldn't be any. So I'd compare those two places. So those pictures look like bar graphs. You have below falls and above falls. These are the places. Everywhere I went was boat access only. Incredible few summers doing this work. I'm going to skip over this, but I've measured a whole bunch of different things. I measured chemistry, so I did chemical analysis. I'll talk about that first. Let's talk about it right now. So this is what I call what's there. Sorry, it's white, it's blinding. We're all like, oh. But this, in a nutshell, I kind of showed already things that we had known before from other work, but it's really nice to see it repeated here, where kind of in the first column you have nitrate, phosphorus, that's the P, and calcium, that's Ca. And lines going up means if there's more salmon, there's more of those things in the soil. Those bar graphs on the far right, the salmon-colored ones are below falls because there's salmon there. So if those are higher, that's what it's showing, is that there's more nitrogen, phosphorus, and calcium in those places where there's salmon. Also found a bunch of things. So the conclusion of this body of work, which was published last year, was that the sites were very acidic. So the pH was about 3.5. That's about as acidic as soil gets. But weirdly, usually acidic soil tends to be very low in what we call cations. These soils were very high. They're also really high in nitrogen and incredibly high carbon contents. And this wasn't just because of the salmon, this was because we have incredibly deep organic soils in these rainforests. But that was something unique amongst the highest carbon contents of the soils in the world. And all those things, though, this is what I argued in the paper, they're all associated with healthy soils. And again, like healthy forests, healthy soils is kind of term of contention right now that we don't really know what it is, but it includes all of these things anyways. And from our agricultural perspective, which is normally how we look at soils, the fertile agricultural things have these types of things, except the acidity. Usually they're around neutral or even basic. Okay, cool. That's the chemistry side. The next question I wanted to answer was, okay, who lives there? So I did a whole bunch of work, the same type of thing, measuring soil. And the important thing here is I extracted the DNA from them. And this is this technique called environmental sequencing, where you take all the DNA, and this is pretty new. It's called next generation, actually, although it's 10 years old now. I think we have like next generation or something. But the end result is this, is you do all this, and you look at the DNA you have, and you compare it to online databases where other people have done the same thing, and you can say like, okay, well, this thing in the database is my data, so that means that thing is over here, right? Cool. Before I show you the results, soil backgrounder. As a soil scientist, in fact, I didn't even know that much about soil before I started, but it's likely the most complex ecosystem on the planet. It's three-dimensional, but still incredibly structured. There's floods and drains every time it rains. Whenever that happens, also the gas conditions. You can imagine things that live there are either drowning or suffering from drought conditions, depending on what happened yesterday. So you have changing oxygen, CO2, even electrical and pH conditions on all different sorts of timescales. There's an enormous amount of heterogeneity in the chemistry, depending on where these soils kind of came from, and also all sorts of physical differences. It's also full of life. And we're just in the very, very early days of learning about this because it's dark down there. It's incredibly variable. And we haven't had the tools to look at it until recently. We've had microscopes and so on, but of course, once you dig them up, the things start dying and so on and so forth. And finally, I'm going to talk to you right now about just these two groups, and there's many others. The soil bacteria are relatively small, and we usually think of them as changing quickly. They live and die, relatively quickly. They're very diverse, and they eat kind of whatever's available. You spill milk on your counter, the bacteria are gonna eat that. Fungi, the tubular creatures, they're networked. Unlike bacteria, they're normally single-cellular. Well, they're a little more, not thread-shaped anyways, they're rounder. Fungi are also diverse, but less than bacteria, we think. They're more permanent, and they're enzyme creatures. They're network creatures. And they specialize in eating things that are relatively hard to get. So you can imagine them breaking down wood, breaking down dead bodies, those kind of things. Eating the bread on your kitchen table. So what did I find for bacteria? Now I'm going to show you guys just some pictures. A lot of different types. I found over 1,750 different varieties. 34% of them I could identify to species level. But 83% I could identify to phylum. So there's a lot of difference and gaps in our knowledge about what is actually there, what kind of species there are. But you can see 42% of them are in this group acidobacteria. Why? Again, because the soils are really acidic. This picture I just think is beautiful. It's almost impossible to interpret. But this is a tree of all the different bacteria that were there. And the darker the color, the more bacteria I found from that branch. I also found some archaea there in the corner, which is one of those groups that I'm not going to discuss. I did find some things about their communities, and I'm not going to explain these too much, but if they're different places on these pictures, it means that those are different communities, different of bacteria living together in those places. So I showed here that salmon are different in shallow soil, which is the kind of gray circles, than deep soil, which is the darker circles. And in the A picture, you can see up on top, kind of around the letter A, there's all those salmon-colored dots. So those are all the places with salmon in them. And so they kind of grouped way off on their own, and everything with less salmon grouped together further down.
So what are the two axes?
Great question. I think they're like NNDS variable axes of something. We can talk about this. We can unpack this. But this is like a PCA, if you know what a PCA is. Too hard to explain really quickly, but I'll be happy to talk to you about it after. The B picture kind of shows the same thing. I think this is really cool. Oh, except here, salmon colored circle would be above the waterfalls, and then gray circles below the waterfalls. So above waterfalls and below waterfalls grouped together, and then they're also separated by soil depth. Cool. So that's awesome and all, okay. So different, if you have salmon, that means the communities are different, so it's changing the environment. But really, I wanted to ask, what are they doing there? So I did a bunch of work. This is the same type of picture. In here, I'm only going to highlight green means that pathway. So these are different pathways of chemistry. Green means more with salmon. So if you just look in like the first picture, the degradation one, right at the top, you have nitrogen deg. That means nitrogen degradation. So this means that salmon increased the nitrogen degrading capacity of the communities of bacteria that were there. That's super cool. This actually I haven't published yet. I just made that a few months ago. So that's bacteria. What about the fungi? Here's a great picture of the fungus. I think this is the Larocque growing through the vertebra there. You can see of a dead salmon. You can also see some rib bones and a fin in the background. And in this case, you can see that the hyphae have been degrading the bones and so on of the salmon. I found about 2,000 varieties. And again, we can look at this later if you're interested in all the different types. And there were many I couldn't identify. So at least 35% were completely unidentified. So that means they weren't in the reference database. So there's a lot of, like bacteria and fungus, there's a lot of things we still don't know that's out there in the world. So I had two hypotheses then about what they're doing. The first was, hypothesis one, if mycorrhiza help plants find nutrients when they're hard to get, which is how we think they work, then if there's more nutrients around, like from salmon, then the plants will need their fungal buddies less. So you're going to have less mycorrhizal fungi. The second hypothesis was that since fungal strands are easier to grow than roots, and this kind of makes sense because they're thinner, right? They're, like, you can imagine, you can see a root, you can hold a root. There's a lot of work that goes into growing a root. Fungal strands are typically more ephemeral, right, in space and time, and so they take less energy. So since they're easier to make than roots, and since salmon nutrients are really patchy, right, and ephemeral in time and space, then you can imagine if you're trying to find these things and you're a plant, you can't move, you need to put resources into exploring the soil in this three-dimensional space. Then it kind of makes more sense for plants to invest in their fungi to do that exploring, which they're really good at, than them doing it, and they're kind of bad at it. So under that hypothesis, the plants will need their fungal buddies more when there's more salmon there. So to look at that then, here I've called the mycorrhizal things symbiotrophs, because they like biology. And the other ones, the other class I'm going to highlight here are the saprotrophs. So these are the things that degrade wood and so on. This is what I found. So first I found there's more diversity of more salmon. That's cool. But in the middle, you can see the green line is the saprotrophs. And so here the x-axis again is more salmon in the first column. So there's more, as you get more salmon, you get more and more symbiotrophs up to a certain point. And then it kind of levels out, or even on the bottom in the deeper soil starts going down. I didn't have quite enough data to really be confident with that, but it seems like that given this hypothesis that we could be seeing there, when you have tons and tons of nutrients, you actually get less symbiotrophs, less mycorrhiza. We kind of saw the same thing then above and below the waterfalls, where with the salmon there'd be more. I think the first column there, I should have included, the first two are the symbiotrophs, and the second two are the saprotrophs. How are we doing for time? How are we doing for energy? Should I be wrapping up?
No.
Are we still here? We can do one more thing. I actually didn't ask how long I should talk for. I should have done that.
We usually go for an hour.
Okay, perfect. So that was neat. So when I first started, I had this amazing, this dream. I'm like, okay, I want to follow the salmon nutrients through the mushrooms and see where they go into the trees. Turns out that's really hard to do, right? But I still wanted to answer that question. So I went back and I did another study. And the question here was, can I show that the salmon, maybe I can't follow like the nitrogen like with a microscope or something. We didn't quite have the technology. But I really wanted to see, is the salmon nitrogen in the fungus? And then can I show any evidence of that then being passed on to the trees? So I used a trick here. And it's called stable isotopes. And salmon, by some fluke of nature, we all have nitrogen in our bodies, right? In our flesh, mostly in our muscles and skin. All of our proteins are high in nitrogen. And it turns out there's two types. There's N14, which is the normal nitrogen, and N15 has 15 neutrons that we call heavy nitrogen, or N15. And it turns out that if you're from the ocean, this is not how it works, but I like to think of it, since the ocean is down, there's more heavy nitrogen that ends up down there. So if you're from the ocean, you have more N15. And also if you're a predator, you kind of concentrate the N15 in your bodies. So you can actually take hair samples and show people, are they vegetarians or meat eaters? So of course, since salmon are both, they're like pretty high in N15. So that's been used by scientists. That's one of the main things when they say salmon nutrients. What they're really talking about is this N15 measurement. So you can take samples of stuff, you run it through what we call an elemental analyzer and do something called mass spectrometry, and you can measure the N15 in these samples. So I wanted to do that for the mushrooms. So I went out, picked a bunch. We have amazing mushrooms on the table over here. So this was actually, for me, my first serious mushroom foray. I did an okay job. But here I brought them all into the lab, and I really wanted to identify them to species as well as I could. I guess I had 170 samples of mushroom litter and soil. And then I measured their N15. So here I grouped them by genus, and it's a little hard to see, but in the red circle there is the Lactarius, Hebeloma, Leucaria, Cortinariuses. The Hebelomas in particular, these are salmon that, they're mushrooms that we call aminophilic, or sometimes we call them corpse fungi. So we know from other studies, and weirdly, those fungi we know a lot about because, of course, forensic scientists study them because we're interested in finding corpses in the woods when they're there. So it turns out those mushrooms in particular had higher N15 concentrations when salmon was there especially, than others. So that's not a slam dunk yet, but for me, I'm like, okay, that's getting some pretty good evidence here that the mushrooms are taking up the salmon. So I wanted to show, though, let me see at the top. Can I show them that the N15 is going from the mushrooms to the trees? So again, I'm going to present to you two hypotheses. The first is that, in this picture, the x-axis is my hypothetical salmon density. The brown line is the N15 below ground, and the green line, because trees are green, is the vegetation, or the above ground. We just call it the tree N15. So since salmon are high in N15, then as you get more salmon along that axis, they both go up together, because the salmon nutrients are just getting dumped in and it's ending up in everything. It's ending up in the mushrooms and the soil as well as the plants. But something weird that we know is that mycorrhiza, they also affect this N15 distribution. And this has created a whole lot of confusion in the literature and debate, because if we're saying the N15 is the salmon nutrients, well, it's not quite, because it turns out mycorrhizal fungi also will hang on to it, and the nitrogen they give to trees tends to be depleted in this N15. So in that case, we have the x-axis your mycorrhizal transfer rate. As it does that more and more, you expect the fungus is hanging on to the N15 and giving the depleted lower N food to the trees. And so you expect those lines to diverge as more and more of that process is happening. So these were kind of like the two processes that were going on, the salmon and the mycorrhizal transfer rate. And I wanted to see which of them, are they both happening? Is one changing the other? What's going on here? Can I say anything about that with the data I had? So here is my two hypotheses then. The whole time, as I get more salmon, I expect that both lines to go up. But if that also causes more mycorrhizal transfer, then as I get more salmon, I expect both lines go up, but also to diverge a little bit. If there's less mycorrhizal transfer rate, then they'll both go up, but they'll be kind of converging as they go. So I hope that makes sense. What did I see? Well, you guys tell me here. The pink line, this is work actually, I'm still working on this and finalizing this. The pink line is the mushrooms, and the brown line is the deeper soil. So that's the kind of below ground stuff. The green line again is the vegetation, and the light brown is shallow soil. So they tended to group together, deep soil and mushrooms, shallow soil and trees. So they do both go up as you get more salmon, and I don't know, they both are kind of diverging. I think this is really cool. I'm still working on the statistics and exploring the best way to kind of, if I can say this is true or not, but in the end, I like looking at pictures a lot of the time, even as a statistician. And if you can look at a picture and say, yeah, that's the thing, then it's probably at least more true than something you have to jump through a lot of hoops to say the same picture, or say the same thing. So my interpretation from this, anyways, my working interpretation is that the salmon nitrogen does go everywhere, but accumulates more below ground than above ground, which is why the below ground's above. And then there's some evidence here, at least, for it increasing in mycorrhizal transfer rates with more salmon around. Someone else is probably going to have to do the next step of that work. So the take-home message from this, salmon do change kind of collectively all sorts of different things in these systems. They change the soil chemistry, which of course will indirectly, other work has shown changes the vegetation that lives there, the animals that live there, the worms that live there, the insects. Salmon change soil bacteria and fungal communities. Those salmon nutrients are taken up by the fungi and end up there. And there's some evidence then that those nutrients are moved into the plants and that it changes the amount that it's getting moved because there's more nutrients around. So for me, getting back to this story of old growth and history, because all of this, of course, were in old forests, right? Scientists, again, one of our blinders is we know a lot about really, really old forests because you get funding to send students like me up to do this kind of work. And we also know a lot about really young forests, things that we've just clear cut because we're really interested in what is the effect of clear cuts. Or then we know a fair bit about trees growing up to, in BC, 60, 80 years old until we cut them down again. We especially don't know a lot about trees. It's actually a massive gap in our knowledge that are between that 80 and old growth stage, whatever that means, 300 or whatever. So for me, that story, that condition, that's a story of history, right? That is, what happened 80 years ago really influences those things. And if old growth trees and old growth stands we're going to preserve, and there's been many arguments made for this to keep them as kind of scientific, things to compare to, right? And that's really, really important that we do that. But it's really important that we do that we preserve the connections that are there, but that we also, I think, preserve and look at the trees that maybe aren't exactly old growth. And again, I think we really get caught up with these definitions. But for me, the trees that are 100 years old, 120 years old, whatever, are just as interesting. And perhaps as those connections between these systems kind of regrow and reform, I think those are really interesting questions. And they're really interesting questions both as a scientist and a person. They're really important as foresters in our society, and how do we, take care of our forests in ways, especially as they're under stress and the world is changing and they don't have as much water to drink and they are getting attacked by all sorts of novel organisms that we're bringing in from all around the world and all the rest of it. So there's far too many people to thank, but I want to thank all of them. And I want to thank all of you for listening to me go on for an hour now. And it was an absolute pleasure to come talk to you. And I'm happy to take some questions if we have time for that. And here's my email up as well. If you guys, again, have opinions to say about forest health or the lack of it in particular, or anything else I talked about, feel free to write me.